Good morning, students. Students, as we were discussing about the tools of recombinant DNA technology, and in my previous video, I discussed about restriction enzymes. So, in today's video, I will discuss some other tools which are required while we are going for rDNA technology. The first one tool, restriction enzyme, as we already discussed, it is used to cut the DNA segment at a particular site. While the second tool is called desired gene. Students, I want to tell you that we will require a desired gene, means a gene which is having our desirable characteristic feature. Means suppose there is a plant and in that particular plant I want to introduce a specific kind of character. Means this specific character will be holded by a particular gene. So this gene is of my requirement. So I have to find out that particular desired gene. So this is the second tool which is going to be inserted into the host DNA. So the desired gene is the second important tool for the rDNA technology, and the functional or normal gene of our interest, it is considered as the second tool, and it is taken from a donor cell. This desired gene is also called as foreign gene, which actually it is taken out from the other plant or other host, so that's why, or other donor, so that's why it is called as foreign gene, or sometimes it is also called as transgene. So this is the second tool, means the second tool is desired gene. While I am talking about the third tool, and the third tool is a kind of polymerase enzyme. As we all know that polymerase enzyme is responsible for the polymerization, means its role is basically it helps in the union of various nucleotides and formation of a long polynucleotide chain. So there are three types of polymerase enzymes which are also very helpful or useful or important for the rDNA technology, and these polymerase enzymes are: first one is DNA polymerase first, second is DNA polymerase second, and the third one is DNA polymerase third. So third tool is DNA polymerase enzyme, and we know that what is the function of DNA polymerase. It is useful in the polymerization reaction, means it helps in the formation of a polynucleotide chain of DNA. While now the third important enzyme or third important tool is called DNA ligase. Now it works opposite to the restriction enzyme. While the restriction enzyme is used to cut the DNA, while the ligase is used to join the DNA segments. Now this enzyme is a very important enzyme, and it joins two sticky ends of DNA, and it is called as ligase. It is used as a molecular स्टिचर मीन्स स्टिचिंग का काम करता है ये लाइगेज जो है जॉइन करने का काम करता है. सो दैट्स वाई इट इज़ कॉल्ड एज मॉलिकुलर स्टिचर्स. ठीक है. डीएनए लाइगेज वाज इनिशियली डिस्कवर्ड बाय हरगोविंद खुराना. ही वाज an Indian scientist, and Dr. Hargobind Khurana was born on 9 January 1922 at Raipur, Punjab, which is now in Pakistan. ठीक है. And he died at November 9, 2011. ठीक है. And he was awarded Nobel Prize for his various works, as we discussed earlier in the sixth chapter, molecular biology. He was the scientist who initially proposed the concept of the genetic codon, and he received Nobel Prize too. So I hope so you understood what is the role of DNA ligase enzyme. DNA ligase enzyme is used to join two sticky ends of DNA. Now I am talking about the fourth one and the last one, very important tool of the rDNA technology, and this tool is vector. And what is vector? I am going to tell you that vectors are the things, or these are the objects, or these are the living organisms which carry DNA. ठीक है. Which carry DNA to the host cell. Means I mean to say that suppose I have to insert one gene of Mohan into Sohan. ठीक है. I want to insert one gene of Mohan, and I want to insert this gene into Sohan. But while I am going to insert the gene from Mohan to Sohan, I will require one intermediate, or I will require one carrier. Suppose Rasmi is carrying that particular gene to Mohan to Sohan. So here Rasmi is a vector. So similarly, there are various vectors which are used to pass the desired gene or transgene or foreign gene from donor cell to host cell. So these are called as vector. ठीक है, these vectors are referred as carrier DNA that act as a vehicle to carry desired gene from host cell to the vector cell. And there are various important vectors which are used in genetic engineering, and these vectors are plasmids, macrophages, plant viruses, animal viruses, sometimes cosmids, and sometimes artificial chromosomes. So there are 6 different types of vectors which can be used in DNA technology. The first one is plasmid, the second one is phases, third one is plant viruses, animal viruses, cosmids, and artificial chromosomes. Now I want to tell you that we have to discuss about only plasmids. Now what are plasmids? I am going to tell you that these plasmids are extracellular chromosomes, means extra chromosomal DNA, we can say that. Here you are observing there is a bacterial cell which is having green colored cytoplasm, and you are observing red colored chromosomal DNA. Except it, you are observing 3 small circular or spherical structures are there. These spherical structures are extra chromosomal DNA. These are also DNA, but these are in spherical shape and these are called as plasmids. ठीक है, these plasmids can be used as a vector which can transmit the foreign gene from a host cell to a from a donor cell to a host cell. Now, what are cloning vectors? I am telling you that the plasmids or bacteriophages are basically used for the cloning vectors. Why? There is a reason behind it. Because they have ability to replicate within the bacterial cell. Okay, suppose there is a bacteria from where I want to introduce a gene into another bacteria, and this bacteria is called Escherichia coli. So I will use definitely I will use a plasmid, ठीक है, as a vector. And why I am choosing a plasmid? Because this plasmid has capability. While I will introduce this plasmid into Escherichia coli, which is a host cell, it has capability to replicate inside the Escherichia coli. So it has capability to replicate inside the host cell. So that's why we are using cloning vectors, because they have capability to make their clones. Now एक जो cloning vector है, it must have some स्पेसिफिक कैरेक्टरिस्टिक फीचर्स. तो यानी कि वेक्टर्स के पास कम से कम 3 कैरेक्टरिस्टिक फीचर्स का होना एसेंशियल है. ना व्हाट आर दीज? द फर्स्ट वन इज ओरी. ओरी इज कॉल्ड एज origin of replication, and we know very well that this is a sequence of DNA from where the both of the DNAs are going to be open and the process of replication starts. And this is the same site where the where we can say that the desired DNA can link. So the foreign DNA can link on the ORI site only, and from here the origin of replication initiates, or the process of the replication begins. Now the second important feature is called as cloning sites, and cloning sites are basically, we can say that these are the restriction sites or these are the recognition sites. Means these are the sequences of the DNA which can be identified by the restriction enzyme, and you know very well that these are the palindromic sequences. ठीक है, एंड द वेक्टर कंटेन्स अ रिकॉग्निशन साइट और पैलेंड्रोमिक साइट्स, ठीक है, विच आर कॉमन फॉर restriction endonuclease enzyme, and from where initially the restriction enzyme will cut the vector here in the side, and finally the same restriction enzyme will cut the host DNA, and the piece of DNA will be stick because they both have the same sticky end. Either I will talk about the plasmid, it will have the same sticky end. Similarly, I will talk about, if I will talk about the donor cell, it will also have same sequences or same sticky end, so it will join very quickly. So cloning sites must be there. And last one is, which is a very important feature for vector, it is called selectable markers. So selectable markers are also those sequences which help us to identify uh, which helps us to identify a transformant from a non-transformant. Means transformant means either rDNA technology, the after the rDNA technology, why when we are getting a new kind of plasmid, this will be referred as transformant, and non-transformant are those who are not obtained by rDNA technology or which are primitive type. So I will discuss it later on in the next video, what are the selectable markers in detail. So I want to show you one figure here. This is a kind of plasmid, or this is a kind of vector, and you are observing there are various recognition sites, and this is the figure of PBR 322. Okay, it's a vector, and we will discuss it in our next video in detail. Till then, thank you, bye bye, and have a nice day.